All right, so last week we were working on naming compounds, and we will get back to that a little later on this week, but it is something you're going to need to spend some time practicing on your own. Okay, today we're going to go over properties of compounds. So we're going to look at what some of the properties of ionic compounds and some of the properties of molecular compounds are in preparation for the lab that we will be doing on Wednesday. Okay, so on Wednesday, you are going to be performing a lab where you will get about a dozen unknown compounds. Okay, and you will have to perform some tests to figure out what their physical properties are. Right? What are physical properties? What are some examples of physical properties? Color, yeah. What else?
Shape.
Shape could be a physical property, yeah. Especially if it's like crystalline. Crystals, each compound would have its own kind of shape of crystal for sure. What else?
Size.
Size, yeah.
State of matter.
State of matter, then, and that is dictated by other physical properties like boiling point and melting point, whether it's a gas at room temperature or a solid at room temperature. Okay, so things like that are examples of physical properties. We're going to test a few different things. Okay, not necessarily like boiling point or melting point because that requires temperature control we don't have. But we will test some things, and from those physical properties, we should be able to identify the 12 unknowns that we'll have. Okay, just based on the list of physical properties that we'll have for each unknown. Okay, so we'll perform the same set of tests on each unknown compound and then look at their data and go, okay, this one would match up with that. This one would match up with that, because you will get a list of the possible identities. Okay, it's not like you're gonna have to pull the identities out of the hat. Okay, you're gonna know one of the unknowns is copper 2 sulfate, and one of the unknowns is water, and one of the unknowns is table salt. Okay, and then you're gonna look at the properties that you figure out and go, okay, so that's white crystalline solid, and it dissolved easily in water, and it conducted electricity, and it was fairly neutral. That's probably table salt. Okay, things like that. That's how we'll be identifying them, how the lab will go. So today we're going to go over the properties that will help you to figure out what each thing is. Okay, and then after that, we're going to look at what the expectations for a lab report are going to be. Okay, so that you guys have that have an idea of that, so you know what will be expected of you. Tomorrow, we'll do a quiz on naming to start off with, and then from there, we'll go into the pre-lab part of the lab. So we'll have the Chromebooks tomorrow. You'll be able to start getting the lab report partially done, and then on Wednesday, we'll actually go into the lab and perform the procedure and have our data so we can analyze it and do the rest. Okay, so that's kind of the plan going forward. And then Thursday, we'll look at how we do on Thursday's quiz on naming, and if we need to spend more time on that, we will at that point. Okay, so that's the plan going forward. So for today, we're going to learn the properties of compounds, acids, bases, ionic, and molecular. Okay, and we're going to understand how those properties can be used to identify a compound, which, as we said, is going to be useful for your lab design. All right, so all matter has certain properties. Some of those properties are chemical properties. Chemical properties refer to whether or how something reacts with something else. That's a chemical property. I require that something else to discover those. Physical properties are inherent to the material itself. I can measure them without other materials or discover them without other materials except maybe water. If I'm doing solubility, I would obviously need to see whether it dissolves in water or not. Okay? But they're the things that are just inherent to that material. So they would be things like color, density, melting point, whether they conduct electricity, whether they're magnetic, okay, all of that kind of stuff, things that we had talked about here earlier, right? So those properties can be used to distinguish between types of compounds, and then within those types, individual identities. Okay. Questions so far, guys? All right, so for ionic compounds, here's the things that we need to know about ionic compounds. Ionic compounds are typically crystalline solids at room temperature, because they tend to have very high melting points, like several hundred degrees. Okay. Now, does that mean every crystalline solid you see is an ionic compound? does not. Okay, all ionic compounds are crystalline solids, but not all crystalline solids are ionic compounds. Okay, the two are not mutually exclusive. Right? For example, sugar. Sugar is a crystalline solid, but it is not an ionic compound. Okay? Incidentally, two of your unknowns will be salt and sugar, which look very much alike. Okay? And we can't do taste tests because some of our unknowns are poisonous or acidic and would dissolve your tongue. Okay? So we won't be able to tell them apart that way. Okay? We'll have to use some other physical properties instead. Okay? Ionic compounds will conduct electricity when they are in solution, and they are typically, but not always, quite soluble in water. Okay? So what I mean by they'll conduct electricity in water is once they are dissolved, the solution that is made will conduct electricity. Okay? Now tap water can conduct electricity, but that's because tap water has ions in it. Okay? Especially hard water like we have here. It's got calcium and chloride and sulfates and all that kind of stuff in it. So it's got ions floating around, and that means there's some positively charged and some negatively charged particles in the water that can allow the electrons to pass between them and thus conduct electricity. Distilled water does not conduct electricity because distilled water is a pure substance and has no ions floating in it. Okay, does that sort of make sense, everybody? All right, so what we would be looking at here, okay, this would, if we could get like a single crystal of like salt, it would probably look something like this, okay, so it's a crystalline solid. When it dissolves in water, okay, the ions, the sodium, which is positively charged because it's given away an electron, and the chlorine, which is negatively charged because it's accepted the electron, are going to separate, and they're going to float around in that water, in that solution now, and conduct electricity. Okay? If this was pure water with nothing in it, there would be none of these charged particles, and then there'd be no ability for the electrons to pass from particle to particle. Okay? So it would not conduct electricity. Okay? Some ionic compounds can even be brightly colored. It depends on the metal involved. Some metals have distinct colors to their crystals. Okay? Not all, but some. Okay? So that can really be helpful if I'm looking at a bunch of unknowns and I got a bright green one. You know what? The bright green one is either iron containing or nickel containing. Okay? It really narrows it down. All right? If it's blue, it's got copper in it. If it's pink, it's got cobalt in it. Okay? So there's some colors that are really indicative of one material in particular. Okay? Everybody all right with that? Okay, so these I would make sure I highlight or somehow indicate because they're going to be important for the lab on Wednesday. Okay, if something is blue, if I've got an ionic compound, a crystalline solid and it's blue, it's going to be containing copper. Okay, if it is green, then it's likely containing nickel. There's an off chance it could be iron, but I didn't put any with iron in as unknowns. Okay, and for cobalt containing compounds, they can be anywhere from pink to like orangish red. Okay, so they have a bit of a range, but they're generally pink to red, okay, in color. So those ones are going to be important for when we do the lab. Okay, if you see something that's pink and there's something that's containing cobalt in the unknowns, that's probably what it is. Okay. All right, everybody okay with that? Okay, for molecular compounds, they're very different. Molecular compounds typically have low melting and boiling points, so they can exist in any state at room temperature. All right? Water, which is a molecular compound, just happens to be a liquid at room temperature, but carbon dioxide is gas. Okay? They're both molecular compounds. There aren't that many, like let's say solid molecular compounds at room temperature. There's much more gases and liquids, and they typically have lower melting and boiling points. Molecular compounds are not usually as soluble in water. Okay? There are obviously exceptions, like sugar is fairly soluble in water, but oil obviously is not. Okay? When we always talk about how oil and water separate. Okay? That's a molecular compound, doesn't dissolve in water. Gasoline, candle wax, okay? Like stuff like that, they don't dissolve in water. Okay, they're organic, but they're also molecular compounds, and they're not as soluble. Okay, if they won't dissolve, then the solution will obviously also not conduct electricity, because there will be no ions, okay, floating around in it. Would that be a way I could tell sugar and salt apart in my lab on Wednesday? Okay, I'd say that would be kind of the really important one, because they look alike, they're both soluble in water, but the one thing that would be different would be because salt is ionic, it will conduct electricity. Sugar is molecular, it will not. Okay, that would be the way I would tell them apart on Wednesday. Okay. So little things like that, it's kind of those, there's always one single property that's going to kind of give something away. Sometimes it's easy, like color, and sometimes you got to go all the way to the pH test before you figure it out. All right, so like we were saying here, okay, molecular compounds, they don't break up into the charged particles, okay, whereas ionic compounds will, and that's why they conduct electricity, but molecular compounds do not. Okay. All right, and molecular compounds typically, when they're soluble, create clear solutions. There really are not as many brightly colored organic or molecular compounds, and they're typically going to just be clear. All right, now for acids, in our unknowns on Wednesday, we're going to have one strong acid and one strong base. Okay, everything else is going to be somewhere between like 8 and like 4. Okay, so like in terms of pH, we're not going to have many things that are extremely basic or extremely acidic. Most stuff is going to be around neutral. Okay, but we're going to have a couple. If something is an acid, a strong acid, it is an ionic compound. Okay? There are no molecular compounds that are strong acids, because in order to be a strong acid, you have to be able to break up into positively charged hydrogen ions and something else. Okay? So if something is a strong acid, it's going to be an ionic compound with hydrogen acting as the metal. Okay? So if I had this stuff, would that be acidic? It would be. Okay? Hydrogen is listed first, so it's acting as a metal. It's an ionic compound. Okay? And when it gets into water, it's going to split into hydrogen and chlorine. Okay? This is hydrochloric acid. It's the stuff that's in your stomach that helps you to digest your food. Okay? Which would be stronger, do you think? Hydrochloric acid or sulfuric acid, assuming they're the same concentration? Okay, why? Okay. Could this stuff also do that? Yeah, it could. Here's what determines whether an acid is really strong or really weak. Because we all are very familiar with hydrochloric acid, and we're like, yeah, that's a strong acid, so we automatically kind of go with, oh, that would be really strong. But what actually makes an acid stronger is how many hydrogens it can give away. Okay? If there's this in solution, it's going to be acidic. The more of that gets into solution, the stronger that acid will be. So this stuff would give away 2 hydrogens, where this would only give away 1. Okay, so sulfuric acid would be even stronger at the same concentration. I could make hydrochloric acid stronger if I made it more concentrated than this. Yeah. Okay, what about this stuff? Stronger or weaker? Stronger. It's got more hydrogen, right? When that goes into solution, it's going to give up 3 hydrogens instead of 2, or instead of 1. Okay? The more hydrogens it can give off, the stronger that acid is going to be. Nitric acid is very dangerous because it can get into a gaseous, like as a fume, not so much a gas, but become a fume. And if that fume touches your hands, your hands will blister. You'll get a chemical burn just from holding your hands near an open container of this stuff. Okay? Imagine what would happen if you accidentally inhaled it. Okay? So if you were near it and breathing and inhaled it, it would do the same thing to your lungs. Okay? It could be very, very bad. Right? What about this stuff? Would that be a strong acid as well? It's got 2 hydrogens acting as a metal, right? So it's an ionic compound. It goes into solution, it's going to give up 2. This stuff is something, if you get into anything to do with oil field stuff, like if you're working on the rigs or anything like that, okay, you have to take an H2S certification for safety, because this is a gas that is often associated with petrochemical deposits. And if it gets out and you inhale it, it can burn your lungs. It's a really big deal. You have to have special protective equipment if this stuff were to escape. Okay, so that can be something that can be very dangerous because it is highly acidic. Okay, is that making some sense? So if you've got an unknown and you do a pH test and it pops like it's a pH of 1, then I would go through my unknowns and start looking for a compound that starts with H. Okay, does that sort of make sense? It starts with H and it's not water, which is obviously neutral. Okay, then that would probably be your culprit. All right, so acids are compounds that have H plus ions in them. Okay, the presence of that is going to give it its acidic properties. Okay, can we have weak acids? Yes, weak acids are typically molecular compounds. Citric acid, for example, is a weak acid. It's a molecular compound. Okay, yeah, stuff like that. Does that make it a little more sense? Now, it says on here, acids taste sour or tart. Are we going to be testing that? We are not, okay? Because when I make up the stuff, I make it uber concentrated because I like to get really good results. All right, so if you get anything on you, you're going to have to like rinse in the sink for a few minutes. Okay, so yeah, we don't want to taste any of this stuff. It will be the last tart, sour thing you ever taste. Okay, because you won't have a tongue after. Yeah. So don't taste any of it, please. Okay. But you've all experienced tasting things that are somewhat acidic. Lemon juice is sour. Okay, vinegar is tart. Okay, those are both acids. Okay, weaker acids, but acids still. All right, and they will turn indicators to acidic colors. We have Hydrion paper, which does the full range from 1 to 14. Okay, the acidic colors become orange to red, okay, as we go along. And there's a color chart. I'll show you what it looks like, okay, that you can use to do that. Okay, bases go the opposite way. Okay, so bases contain hydroxide ions, so a polyatomic ion. Right? So if I've got a compound and it's ionic and it ends in OH, like this stuff, that would be a strong base, sodium hydroxide. Okay? Sodium is the metal, hydroxide is the nonmetal. Whoop. If I put this into solution, I will get a strongly basic solution. In fact, this is what's in oven cleaner. Okay? So if you've ever sprayed the foaming stuff, okay, inside your oven, okay, that's what's in it. Right? That's a strong solution of sodium hydroxide that can basically dissolve the most burned on crud that is in there. Right? But it's also why you are not supposed to be near it when it's working. Okay? Why you keep the oven closed and all that kind of stuff. All right, so anything that is ionic and ends in OH is going to be a strong base. Which would be stronger, this or this? Assuming equal concentration. The second one, it gives off more OH. The more hydroxide that it can put into solution, the stronger it's going to be. Okay. All right, this stuff here, this is lye. Okay, it's the stuff that's, well, you put it in like outhouses and stuff to kill bacteria and keep the smell down. Right? Does a good job of that. Okay. All right, so basic solutions are going to have a pH of higher than 7. Okay, so 8 through 14. The stuff that we're going to have that's going to be a strong base in our lab is going to be 13, 14. Okay, it's going to be really, really strong. All right, and acids typically taste bitter. Right, so soap, for example, okay, tastes bitter. It's got bases in it. Okay, it's typically a basic material. Lots of like medications and stuff will taste bitter because they've got basic compounds in them. So if you've ever like accidentally chewed like a Tylenol or something, okay, it tastes really, really bitter. Okay, questions on acids and bases? Okay, so if I get something and it's a really strong base, I should be looking through my table of unknowns, looking for something that ends in OH. Okay, would this be a strong base?
Yes.
Okay, is it ionic? Does it start with a metal? It doesn't. So this is not an ionic compound, right? Which means when I put it in solution, is it going to break off? Is that OH going to come off? It's not. Okay, this stuff is not a strong base. This is alcohol. Okay, ethyl alcohol. It's what's in like beer and wine and whatever else. Okay, it's not a strong base. In fact, it's mildly acidic. Okay, it's not an ionic compound, so when it's dissolved in water, and it is soluble in water, okay, this doesn't come off. So it does not create a basic solution. If it was an ionic compound, like these, the OH and the metal would separate. Okay, this won't separate, so it won't be basic. Spoiler alert, this is also one of your unknowns. Okay. All right. Okay, so that's what you need to know for properties. Okay, so like I said, the way this is going to set up is you're going to have your unknown compounds, and there's going to be some tests, simple tests, that we're going to run. Okay, and we'll talk about what those simple tests are here in a minute. First, I want to talk about lab report expectations. So, in Google Classroom, one of the documents I included in Google Classroom in the classwork tab. Okay, so worksheet packages and other materials, you'll see one of the things in there is lab report expectations. If you can have that open to follow along with, okay, that would be great, because we're going to go over what your lab reports are going to look like. Okay, so here at HTA we have a lab report template or format that everybody follows. Okay, so after you take Science 10, it'll work for chemistry, physics, and biology, because everybody uses the same one, so you don't have to learn 3 different ones. Okay, our expectations are that you can write a problem, that you can identify variables and explain them, so you're manipulated, responding, and controlled variables. Okay, that you can interpret, write, and or follow a procedure, that you can accurately collect and record data, that you can analyze that data, sometimes with a graph, sometimes just kind of anecdotally, it's whatever is appropriate for the data you have, whether you could draw a diagram, okay, of your data, stuff like that, and then perform some analysis, draw a conclusion, and find sources of error within the lab. Okay, that's what would be expected of you on a typical lab report. Okay, so what you get from me at least, okay, in terms of getting a lab, is if I give you a lab, you'll get something like this. So it'll show up in Google Classroom as labs and assignments, and it'll have a name. Like on Wednesday, there'll be one that says Properties of Compounds Lab. Okay, if you click on that, you'll see a few things. All right, you'll see a PDF document, which is the lab sheet that you would like follow, or that you can kind of use to know what the procedure is. Okay, sometimes there'll be included data. Sometimes it's a document, and sometimes it's a video. For the case of this lab, it's a video, but you're not going to need to use it because you're actually going to do the lab. I only put the video in there for people that are absent, or when they look at their data, it doesn't make any sense, and then they need to use mine. Okay. And then there'll be another video where I'm going through the expectations and explaining the lab. That one's a good one to watch or listen to while you're finalizing your lab report, because I typically give lots of big juicy hints, okay, while I'm outlining the lab. Like I give away some of the variables, or give big hints for what I'm looking for in the hypothesis, or tell you how to do the analysis, stuff like that. Okay. And then there'll be a Google Doc as well, okay, that you will hand in, okay, that you'll submit on Google Classroom. So when you get that, okay, it'll have the skeleton of the lab, and you just have to fill it in. Okay, so it's already set up in the right format. You just have to type in a problem, type in your variables and explain them, make a hypothesis. Okay, this is what you would submit to me on Google Classroom. Okay, all right, so here's what goes in all those blanks. Right? For the problem, the expectation is that you write it in the form of a question. All right, so let's say we're doing a very simple lab where we want to find out what the effect of light is on plant growth. Obviously, that's very simple, okay, but let's just say we're doing that for the sake of having something simple to do. All right, so our problem would then be, what is the effect of light on plant growth? Okay, that's the problem we're investigating. In the design part, you are going to be expected to identify and explain the manipulated variable, so the thing that you as the experimenter are changing to test the effect of. The responding variable, the thing that you as the experimenter are watching, recording, okay, and being aware of any changes in. And then 3 controlled variables. Those are things that have to be kept the same, because if they're not, they'll ruin the results. They'll affect the experiment. Okay, so that's the other thing that has to be listed there. So a manipulated variable, there's only ever one. Okay, responding variable, sometimes there can be more than one, but more often than not, it's just one. And 3 controlled variables. There's often more than 3, but I only require you to identify and explain 3. Okay. All right, so let's say we're doing this lab on light and plant growth. And this is the way I set it up. I'm going to have 4 plants. One plant is going to sit in total darkness 24 hours a day. Okay? That would kind of be my control plant. Okay? Because I have to know what happens if the plant gets no light. Spoiler alert, it's going to die. Okay? Then I'll have a plant that's going to get 6 hours of light. A plant that's going to get 12 hours of light, and a plant that's going to get 18 hours of light. Sorry, I need 5 plants. And one plant that's going to get 24 hours of light. Okay, so it's going to be in the light constantly. And they're going to be right beside each other in a room, and all I'm going to do is close off the boxes so that the light doesn't get to them. That way everything else is the same. They're going to get the same temperature. They're going to have the same air. I'm going to water them the same amount every single day. All right? They're going to grow in the same soil, the same amount of soil, the same type of soil. All of that is going to be kept the same. Okay? Because otherwise the experiment won't be a controlled experiment. Everyone follow me there? Okay. So in this experiment, what's my manipulated variable?
The light that the plants are getting?
Right. And specifically, the amount of light that the plants are getting would be my manipulated variable. So that would be the first thing I have to do, is identify. Okay? In this experiment, the manipulated variable is the amount of light the plants are getting. Okay? Then I have to explain how and why. Okay? So the how part would be every plant will be in its own box, and the box can be closed off to the light so that the plant is shaded for certain periods of time. Okay? This will allow us to find out what the effect of light exposure is on the rate of a plant's growth. Okay? So the how and the why. I'm doing it for this reason, and this is how I'm doing it. Everyone follow? Okay? So for each variable, you're going to have to write, a sentence or two in explanation. What's my responding variable? The growth of the plants. Okay? Now, I would probably want to specify what measure or measures I'm using to record the growth of the plant. Okay? So we could say the growth of the plant is the responding variable, and it will be measured by measuring the height of the plant and counting the number of leaves it grows over a 10-week period. Right? So have I explained how it's responding and why it's responding? Sort of. Okay, I certainly said how, here's how I'm gonna record it. Okay, why is it responding? Because plants need light for growth, and the more light they get, the more they should grow. Okay, all right. What are my controlled variables? They only need 3.
The water.
The water. Okay, I'm gonna water them all the same amount, and with, I would probably specify tap water from Okotoks or distilled water. Okay.
The soil?
The soil, okay? They all need to get the same kind and same amount of soil, because soil is where plants get their nutrients from, okay? And that needs to remain the same. So have I explained how and why I'm controlling both of those so far? Okay?
Same type of plant?
Same type of plant. Yeah, it would hardly be fair to plant you know, a strawberry, a string bean, and a giant sequoia, and then like compare them. I mean, they have very different rates of growth, okay?
You want the same pot, same sauce?
Yeah, same pot, same size. Yeah, you gotta make sure everything that could otherwise affect the growth of a plant has to be kept the same, because I only wanna know what the effect of my manipulated variable is. Okay, does that make sense? So that would be the first 5 marks out of 30, okay, for a lab report, is identifying and explaining your variables. The thing people forget to do is explain. They're very good at identifying, but they don't always explain. Always explain your variables, how and why. Okay, so what you can see here, okay, so I've said in here, the manipulated variable is the part of the experiment changed on purpose by the experimenter. You wouldn't obviously have to write that. Okay, for example, the amount of light given to plants is the manipulated variable because all subjects will receive different amounts of light, and light powers photosynthesis. Okay, for my responding variable, okay, we've already said that's what changes as a result of the manipulations performed. Okay, so I would say the growth of the plants is the responding variable because plants getting more light have more energy for growth. And I would indicate I'm measuring it by these measurements: number of leaves, height, maybe I take pictures of the plants every day, something like that. Okay. And then my controlled variables, they are the other factors that could affect the outcome of the experiment were they to be left unchecked. Okay. For example, same species of plant because different plants grow at different rates. Same amount of water because water is an important factor in plant growth, so each plant needs the same amount so it can't affect the growth rate. Also the same type of water. Okay? Same soil because plants get nutrients from the soil. Different soils hold water differently. Keeping them the same ensures it cannot affect the experiment. Same temperature. Plants grow typically slower in extreme heat or cold, so it will be important to make sure they all have the same temperature. Okay? So I identified 4 there. You only have to identify 3. Okay, all right. What 3 words need to be in a hypothesis? If, and then. Those 3 words must be in a hypothesis. A hypothesis does this. Okay, lots of people, they come into grade 10 like, a hypothesis is a guess. Nope. Okay, guess is a guess. A hypothesis is a statement. Okay, and a hypothesis says this. It tells me what the relationship is thought to be. It tells me briefly how it will be tested, and then predicts the results and tells me what I can do with them. So it has to do 3 things. You can just go fetch that, whatever it was. Okay. All right, so it's an if, and then statement, okay, that communicates the relationship between the manipulated and responding variable, so that's the if part, okay, briefly describes the experiment, that's the and part, and outlines the expected results, that's the then part, okay. So if I was doing an experiment on the effect of listening to music while studying on test performance, okay, my hypothesis might go something like this. If music helps to focus the brain during periods of academic stress, and an experiment is run where some students listen to music while studying and some don't, then on a prescribed test for all subjects, those students who listen to music would perform better. Okay, so I've said what I thought the relationship was. I said I thought music was going to help people study. Okay, it was going to help people learn. I said how I'm going to test it. I'm letting some listen, some not, and then I'm testing them all. Okay, and then I predicted what my results would be. Okay, that's what you do in an experiment. And the then part is important. You have to take a stand. Okay, you have to say this is what's going to happen. You have to be set up to be right or wrong. You can't sit on the fence and go, well, I thought it would be some of each. No, okay, it's one or the other. Okay, and it's okay to be wrong. Okay, I always have people that like come in and they're like, but I think that hypothesis will be wrong. So, you're not going to lose marks if at the end of your experiment your conclusion says, I reject this hypothesis. That's what happens most of the time in real science. Okay? In real science, most hypotheses end up being rejected. Okay? Then we learn from that and we try something different. Right? In our labs, they're pretty cookbooks, and most of the time we're going to accept our hypothesis because we kind of know what's going to happen already. Okay? But it's fine if you end up at the end of your lab saying, I reject this hypothesis. You're not going to lose marks because your hypothesis ended up being wrong. That's okay. As long as it's in the right format, that's what we're looking for. Okay? Does that make sense? Okay. So for this lab with the plant growth, okay, I might write something like this. If plants require light for growth, all right, I just said the relationship between light and growth that's required, okay, and 5 identical plants are given different amounts of light, that's a very brief description of my experiment, but it's good enough, okay, then the plants that receive the most light will show the most growth. Okay, is there a chance I'm going to have to reject this hypothesis? There is, because I have said the more light, the better. That might not be true. Okay, I might have to reject that. That's okay. All right, so as we said, if, okay, the if part says, okay, the cause of the problem or the relationship and describes the experiment, and then what to expect if you are correct. Okay, and we generally try to avoid the use of I, us, or we in a hypothesis. Okay, it is a statement, and we don't want to involve like the first person, third person kind of stuff. We want to write it as though it's kind of an objective statement that we are separated from. Okay, so the worst thing you can write in a hypothesis is this. I think the plants that get the most light will grow best. This is going to sound really harsh, but I don't care what you think. I care what you can prove. Okay? And in science, we don't say what we think. Okay? In science, we say, if this is true and this is done, then this will happen. Okay? Does that make sense? So never write I think in a hypothesis. All right. For the procedure, okay? The procedure is going to be a detailed, step-by-step, explicit set of instructions for you to follow. In our first lab, you guys are actually going to write the procedure. It's pretty straightforward, and I'll help you with it. After that, they'll all be set up for you. Okay? Now, when you write a procedure, does it have to be really specific?
Yeah.
Yes, it does. Because if it's not, people will make assumptions and they'll mess it up. Okay? So you have to write it as though the person trying to follow your instructions is a complete idiot. Okay? Do not take anything for granted. All right? Like, if you want to make sure that they get, that they plant the plant right side up, basically. Okay? Like, when you plant the plants, put the roots down. Okay? And the green part up. Okay? Like when you're laying sod, the green side goes up. Okay? Like, you really have to be that specific. Okay? And it's not to be insulting to anybody. It's to make sure that your experiment can be replicated. If you don't say everything explicitly, and I don't mean by using 4-letter words, I mean like be very clear. Okay? There's a chance that they won't follow it correctly, and then they won't get the same results. Okay? So it has to be very, very explicit. Okay. After the procedure will be your observations, and your observations can come in a variety of forms. Sometimes they're going to be numbers, because you're going to be measuring stuff. Okay? For the lab we're doing on Wednesday, there's going to be some numbers, like conductivity will be a number, pH will be a number, okay? But things like color and state of matter and stuff like that are going to be written observations, okay? They're things that you can see and you'll have to record, okay, by kind of written word. So observations come in all types. There's not a good or a bad for any of them. They're whatever's appropriate for that particular test. Okay, so observations are important, and you get marks for them. Okay, so your design, that's your variables, 5 marks. Your hypothesis, 5 marks. Your procedure, if you have to write one, is 5 marks, but most of the time you won't have to write that. Your observations are 3 marks. Okay, and then we'll move into the analysis from there. Okay, so your analysis is often just some questions for you to answer on the lab report. Okay, on this particular lab that we're doing on Wednesday, our analysis is going to be figuring out what compound is which. Okay, so A was water, and B was hydrochloric acid, and so on. Okay, that'll be my analysis. So they could be calculations, review and comparisons of data, answer questions, perform calculations, and give detailed explanatory answers. Now, for Wednesday's lab, there's not going to be any explanation. There's just going to be a chart. Okay, and it's going to be A, B, C, D, and then you write the identity beside it. Okay, so that's all there's gonna be. There's not really gonna be an explanation or anything like that. You're just identifying. Okay, for the plant lab, let's say that my observations came back like this. Total darkness plant, dead. Okay, plant that gets 6 hours of growth grew 15 centimeters and had 15 leaves. Okay, plant number 3 that got the 12 hours of light grew 25 centimeters and had 30 leaves. Okay, the plant that had 18 hours of sunlight grew about, let's say, 16 centimeters and had 25 leaves. And the plant that got 24 hours of sun died. All right, those are my observations. If I analyze that data, what does it tell me about sunlight and its relationship to plant growth? Does it tell me that more is better?
The opposite. I
Yeah.
think more is the more better.
More isn't always better, okay? There's a happy medium in there. There's kind of a sweet spot, right? And it's somewhere between 12 and 18 hours, okay? Because my 6-hour plant didn't do so good, okay? It didn't do as well as my 18-hour plant. My 12-hour plant did pretty well, okay? And 24 hours was dead, so I know more isn't better. Okay, the 18-hour plant also tells me that it didn't grow as good as the 12. So there's a certain period of time, a certain number of hours of sunlight that is ideal. That's the analysis that I make from my data. Right? Then I go to my conclusion. Okay? And my conclusion, that's what I state. Okay? I state that my hypothesis was not supported by my data because the 24-hour plant died. The 12-hour plant showed the best growth. So somewhere between 12 and 18 hours of sun is the ideal amount. Further experimentation is needed that can study maybe more detail within that time frame. Okay, that's a good conclusion because it says, here's what my data was, here's what it means, and here's the conclusion I draw. My hypothesis was not supported, and here's what I can do the next time. Okay, everybody all right with that? Okay, so for you guys, on a lab report, what you would do with the conclusion is you would copy and paste your hypothesis from the beginning, just copy and paste it, and then tell me, this is the data, this is what it said, this is supported or not supported. Okay, that's your explanation. Okay, your explanation is, this is what the data is, this is what the data says, and now I can accept or reject. Okay. And then the last thing you have to do in a lab report is identify 2 sources of error and suggest improvements. Okay, so within that experiment, I had boxes and I closed off some of the boxes to light. What else did I close them off to?
Oxygen.
Yeah, I might have trapped gases in those boxes. That means that as the plant carried out the dark reactions of photosynthesis, the oxygen it produced could not escape from the box and could have reached toxic levels for the plant. Okay, not something I had anticipated, but it was certainly a source of error that could have affected the growth of the plant. Okay, so next time maybe they each need to be in rooms that are temperature controlled, okay, but where I can flick the lights on and off. Okay, that would be a good improvement to the lab that would take care of that source of error. So that's what you have to do with 2 sources of error. Find 2, tell me what effect they had and how you fix it. Okay, the best place to look for sources of error are the controlled variables, because you know those controlled variables could affect the experiment if anything goes wrong. So they're a really good place to look. Okay. A second source of error could have been, when the plants aren't under the light source, the temperature might have dropped in the box. Okay, so the shaded plants may not have been at exactly the same temperature the entire lab, and that would certainly affect their growth. Okay, and so maybe next time, again, we go into temperature-controlled rooms, okay, where I can make sure that the heating is independent of the light, okay, something like that. So never feel like you need to worry about finding errors in the lab and hurting my feelings. Okay, it's not a reflection on me. I designed the experiments to have errors for you to find. Okay, so you're not being critical of me. Okay, unless that makes you happy, then go right ahead. But okay, you're being critical of the experiment and you're finding the errors within it. The one thing you don't want to do is admit to human error. Okay, because I have people do this all the time and they're like, yeah, we messed up the pH test for like samples A through C, so those numbers aren't good, and that's the source of error. Well, you're damn right it is, but why didn't you fix it? Okay, like all you did was just say to me, hey, Coderre, I screwed up. I did nothing to fix it, and I know that I screwed up, and here are my results, which are now meaningless. Okay, that is not the thought you want me to leave with, because the last thing I mark on your lab is the evaluation. And if you say to me, hey, Coderre, I was too lazy to fix my mistakes, that is not going to help you. Okay, so if something like that happens, fix it right then. You can fix that. That's human error, and you know it happened, and you can fix it. The sources of error we're looking for are ones that were because of the design of the experiment. You followed the procedure. That's not your fault. Okay? Those were errors inherent to the design of the experiment, and here's how I could fix them. Okay? If you actually screw something up, fix it, do it again. Okay? If you don't follow the procedure properly, fix it. Okay? Does that make sense? And don't tell me about it. Okay? Right? Fix it and don't tell me. All right. Does that make sense? What we're looking for there? Okay, so it's a bit of work. There's going to be some typing and stuff here for these, but typically the first one kind of takes a while because it's really your first real lab report. But once you get the hang of these, they really shouldn't end up being a ton of work outside of class. 20 minutes, half hour at the most outside of class to get a lab report done. If it's taking you more time than that, then you better come in and see me because there's something that's not right. Okay? And I don't want you to be spending 6 hours on a lab report. That's not at all what I'm expecting you to do. Okay? If you've spent like an hour outside of class working on a lab report, there's something you're not getting. You should just come in and ask for help. Okay? Don't spend gobs and gobs of time at home on a lab report. All right, I'm gonna give you guys a little break here. What time is it? Okay, so I'm gonna give you guys, let's say, a 5-minute break within the classroom here, and then we're gonna look at what tests are gonna be important for the lab on Wednesday, and some lab safety stuff. Yeah, go ahead. When you get back. Okay, I'll be right back. I'm just going to grab a couple of things from the lab here to show you. I'm still recording. Okay. Okay, so we're gonna be trying to identify these unknown materials based on just their physical properties. Okay, now, we already talked about what some physical properties are, okay, but we don't wanna have to like test forever and ever. We only wanna have to run about 4 tests or so. Okay, so what's something really simple that we can like just start writing down about the materials without really even doing anything? What they look like, yeah. Okay, we're gonna run what we generally call the observable properties test. That means, what can I get just by being in close proximity to this compound? Right? What can I find out about it? I can find out what color it is. I can find out whether it's crystalline, or I can find out whether it's a solid, liquid, or gas. There aren't any gases, by the way, because they're very difficult to keep in a beaker. Okay? You know, whether it's got any bright colors, whether it has any like obvious odors, okay? Things like that. Those are the readily observable properties that we would want to record about each compound. Okay? Because we know that for some of these compounds, color is going to be that one factor that tells us what they are. Everything else is just going to help confirm them. Okay? So the first test we're going to want to run is our observable properties test. Okay? And that is as simple as telling somebody, make the following observations and record them. Okay? But again, be specific. You're going to want to say, perform an observable properties test where you record the following: the color of the material, the nature of the material. Is it crystalline? Is it waxy? Is it soft? Okay? You know, what other notes can you make about its appearance or texture? Okay? Its state. Is it a solid or a liquid? Okay? Does it have any discernible odor? And how do we test odor? Do we put our nose right over the sample and snort? Okay, we waft, right? And quite frankly, if it's not obvious, it doesn't have a smell. All right, like the ones that are going to be, their smell is going to be important, you're going to know it right away. Okay, like it will have a very obvious smell. If you're like, it probably doesn't have a smell. Also, I thought this was common sense up until like during COVID. If someone has got like a cold and they're congested, they should not be the person doing the smelling. Okay, because I had a kid come up, like, and he's like, oh, I can't smell it, and things like, really? Okay. And I go and smack your group because, man, why did you send that guy? Okay, so yeah, make sure it's always someone who's got a good sense of smell. They're not hard of smelling, or they didn't lose their smell during COVID. Because that does this thing. Some people lost their sense of smell after that. So don't send that person. Okay, send a person who can actually smell the stuff. Okay, up to the front. Don't send someone who's colorblind. Okay, Mr. Van Herb next door, he's colorblind, like totally. Okay, when he retired, we bought him those glasses that let you see color when you're colorblind. He had never seen green. Like you put these things on. Yeah, so don't send, if there's anyone, if you know you're colorblind, like don't volunteer to be the person who goes up front, records the colors. Okay, send someone who can see. All right. So observable properties. Okay, that would be things like color. Okay, state of matter, solid, liquid, or gas. Okay, nature of the matter, crystal, cloudy, molten, soft, waxy, buttery. Okay, odor test. Okay, performed, of course, by wafting the material, not by snorting it. Okay. And this is the easiest test because we don't really have to do anything with the materials except observe them. Okay, another important physical property is solubility, whether something dissolves in water or not. Okay, so when we're in the lab, you're going to get one of these. Okay, they're called the depression plate, okay, because they have depressions in them, not because they're sack. Okay, so you're going to put a little bit of each sample in this depression plate, like enough to cover the bottom. You don't need a big pile, don't fill it up. Okay, we just need enough to cover the bottom. And then, that's when you'll do your observable properties test, okay, do all of that. The wafting test I would do with the original large sample, because if you've got 6 samples in here and you start doing this, you're smelling them all. Okay, so I would do the wafting test with the big sample at the front of the room. Okay, once you've done the observable properties, then you're gonna want to find out whether the substances are soluble or not. Okay, so to do that, you're gonna add distilled water to each material. Something else, so how can I describe whether something is soluble or not? Like what's it gonna look like when I add the water? Yeah, okay, maybe it goes clear. Okay, what happens to the stuff? Yeah, now if it doesn't all dissolve, does that mean it wasn't soluble? Not necessarily. So I need to look at things like, do the crystals shrink? Okay, is there any change in the material? Does the water take on the color of the material? Doesn't mean that it all has to dissolve, because quite frankly, you probably won't get all of it to dissolve in the amount of water that you can put in here. So you have to be telling people, in a solubility test, solubility looks like this. Here are the things to watch for and record. Okay? Another thing you're going to want to do while you're doing the solubility test is you're going to want to use this scientific instrument. Okay? This is called a thermometer. You probably call it a thermometer, that's fine. Okay? So when you add the water, have this in there. If there's a temperature change, that's also an indicator that the material is soluble. Okay? You're going to have a few that'll get hotter and one that'll get colder. Okay, when you do the solubility test. That's important stuff to note, because that can be one of the identifying characteristics, okay, if there's a temperature change during the solubility test. And solubility will tell you a little bit about whether it's ionic or molecular. Okay? We know that generally ionic compounds are more soluble. So if you got something that doesn't dissolve, it's probably not ionic. Okay, everyone with me there? Okay, so you'll just do that, and you can stir it a little bit, okay, and whatever, and just kind of watch it for a little while. What's the little red plastic thing for? It's actually rubber, not plastic.
For you mark where it was before or after?
That's what a lot of people use it for, but it's actually to keep your fingers from interfering with the temperature reading. Okay, because if I'm holding the thermometer like this, I'm warming it up. Even if I just have my thumb and forefinger on it, my thumb and forefinger are going to warm up the thermometer. If I grip it by this, I'm insulated a little bit, okay, from the rest of it. All right, so kind of put it in, and record the initial temperature, and then add the water and see what happens. All the water will be at room temperature, and it will be in these little bottles. Okay, so you just squeeze them to get the water out. Okay, pretty straightforward, pretty simple. Okay, they're not water pistols. Okay, they're not squirting each other with them. Okay, that's a lab safety thing. All right, so since this is distilled water, and so is this stuff that's in here, okay, we should get pretty good data on whether it's soluble or not. Once we've done that, the next thing we want to do is our aqueous conductivity test. Okay, that means we want to test whether the solution conducts electricity. What will that tell us? Don't say whether the solution conducts electricity. Obviously, it will tell me that. What else does it tell me?
If it's ionic or
If it's ionic or molecular. If it conducts electricity, it's ionic. Okay, and by conducts electricity, I mean like you got a number of 5 or higher. You're not going to get too many results in the middle on this. You're either going to get it's full on conductive or it's basically doing nothing. You might get the odd one that's like a 3 or a 4. That's essentially non-conductive. Okay, so sorry, I forgot to put the solubility test up. Okay, so for solubility test, record whether it's dissolving, a temperature change, a color change, an odor change, or anything like that. Okay, are the things you want to watch for. Okay, so the conductivity testers are easy to use. Okay, there's a little button on the side of them. Okay, you press and hold that, and the little on light comes on. Okay, it's a little red LED thing that comes on when they're on. And then I place it in the solution. All right, you can see this is distilled water, and it's showing me that it's basically non-conductive. Only one bar is lit up. Okay, so for distilled water, which is obviously a molecular compound, okay, I should get that number. I should get basically non-conductive. Okay, everyone follow me there? All right. Now, if I take that distilled water, okay, so this is about 2, so it's similar, okay, and if I add something to it, like salt, is salt ionic or molecular? It's ionic, okay. All right, so I put some salt in there, and it's going to partially dissolve. Okay, should I get a different result now? Okay, like it's what? It's so conductive that it lit up the last 4. Okay, that's usually a sign that it's like soup. It's more conductive than it can even read. Okay, so that's a 10. Okay, it's highly conductive. If yours does that, though, I'll get you a different one because it really shouldn't do that. Okay, everyone with me there? That's a pretty simple test, right? All you have to record for that test is the number on the conductivity test. Okay. All right. And then, okay, so that's the conductivity test. So have you write this down here. All right, so you just look for the lights. And how important is it for you to rinse anything you stick in those solutions in between tests? It's super important, okay? If I take the conductivity tester from salt water, which definitely is ionic, and put it in the next thing, do I bring some of the salt over? I do, and that's going to contaminate my next solution and give me bad data. So I should rinse it. Tap water or distilled water? Distilled. Spray it with your spray bottle, okay, in the sink. Okay, spray it off between tests. Same with the thermometer. Spray it off with distilled water between tests so you're not carrying any ions from one sample to the next. Okay. And then your last test is your pH test. So we're going to use the Hydrion paper. Okay, it comes in these little tubes, and you can see there's a color chart on the side. All right, so the Hydrion paper is just this orange stuff, and one strip will do 2 samples. Okay, so just tear it in half, and all you have to do is dip it in, just get it wet, okay, and then you hold it up to the sample thing here. So it's showing a pH like 6, right? That's pretty good. It should be around 6. It's not quite green enough to be 7, okay? But I mean, it's just salt and water. Salt doesn't make water like extremely acidic, okay? And then we're good. All right. So does that make sense for how the pH test works? Why do we have to do this test last? Like the order I showed you is the order we have to do the tests in. Why would we do it last?
Do the pH strips leave stuff for water?
Exactly, yeah. Once I put the pH strips in there, I cause a chemical change. Okay, and so there could be stuff now in that solution. I can't perform any other tests and have them be valid after that. Okay, and obviously I can't test the solution's conductivity until I have a solution. That's why I do this one after the solubility test. Okay, and why I do the observable properties before I put water in there. I can't really observe what it looked like once it's in the water. Okay, so there's a specific order that we have to do the tests in. Okay, all right. How are we feeling about these tests? Okay. Are they pretty easy? Pretty straightforward. All right, so each group will have all of that material in the lab on Wednesday. Okay? And then when we're done, obviously we clean everything up. Now, in terms of lab safety and things like that, okay, what's the first thing you do when you get into the lab? Yeah, I'd say wash your hands, not a bad idea.
Are there safety goggles?
Yes. Put on the safety goggles. In the top drawer of each lab station, there should be sufficient goggles for each station. Okay? If your station doesn't have enough, we'll find some more. But everybody needs to wear the safety goggles. If you wear prescription glasses, those are not sufficient on their own because stuff can come in over top or underneath and from the sides. Okay, so if you wear glasses and you don't have contacts, we'll give you the really big safety goggles that fit over top, okay? And they leave these nice marks on your face. And you're lucky, you get me second semester, this always seems to happen on picture day first semester. Okay? And it's not because I plan it that way, it's just that it always seems to work out, people get the nice goggle marks in their pictures. Okay, so we've got good goggles, like if you've got contacts, the normal safety goggles that we have are lightweight plastic, they're easy to see through. Okay, but if you have glasses and you have to wear them, then we'll get you the big ones that go over top. They're a little less comfortable, but it's more comfortable than being blind. Okay, so everyone wears the goggles until I say to take them off. Okay, and then there's lab aprons as well. You don't have to wear the lab aprons, but if you want to, you can. Okay, I would suggest not wearing your favorite clothes on lab day. Okay, stuff happens, okay, and I wouldn't want you to burn a hole in, okay, or something like that in your favorite sweater or something like that. Okay, so yeah, wear something that's, you know, maybe not your favorite, and make sure it's not like really baggy or has parts that hang out. So if you've got a hoodie on, tie the drawstring and tuck it inside. Okay, if you've got long hair, your science binder should have a hair tie in it. Okay, rubber bands, something to tie your hair back, because there's nothing worse than suddenly I smell burnt hair, okay, in the lab because somebody's hair got in the acid, okay, and now it's all crinkled up and looks terrible and they have to go get a haircut. Okay, so have it tied back. Okay, if you got long hair, tie it back. No hats in the lab. Okay, you'll have to take the hats off for the lab. Okay, that's just a safety thing. And horseplay. If anybody is fooling around in my lab, you're done. Okay, you're coming over here and you're not doing the rest of the lab, and I'll consider whether you're doing any labs after that. Okay, I take safety very, very seriously. So do not horse around in my lab. No pushing and shoving, doing whatever. Okay, you're just doing a lab over there. That's all that should be happening. Okay, if you get something on you, okay, just on your skin, not a big deal. Just go to the sink and rinse it for 5 minutes under cold water. Okay, that's all you have to do. If you get it in your eyes, you weren't wearing your goggles. So wear your goggles. Because when you get it in your eyes, it's really unpleasant for both of us. Because you're going to be thrashing, maybe screaming, and I'm going to have to take you to the eye wash station. And you're probably going to fight with me while I'm doing that because you'll be very panicked and it's not fun. And so what I usually end up doing, because I've had to do this, is like I take a handful of your hair and I just take your face and jam it into the eye wash station. It's not fun for either of us, okay? And so you're thrashing and kicking and screaming and I'm holding your face in and it looks like I'm trying to drown you, but I'm not, okay? Because the little eye wash thing just shoots, it's like a water fountain except it comes from both sides. Okay, and it gets in your eyes and it rinses out your eyes. Okay, but it's not fun because I got one handful of hair and I'm peeling your eyelids back with the other hand. Okay, it's not fun. Okay, and the last time it happened, both me and the person ended up bloodied. Okay, yeah, because like they were thrashing and it wasn't their fault. They were in pain, they were scared, and whatever, but they went and they backhanded me right across the nose. Okay, and that wasn't fun. And then I'm like, and I got them right across the nose on the eye wash station because there's a bar that goes across. It was ugly. So can you just keep your damn goggles on so we don't have to have a repeat of that? It was not cool. Okay. So yeah, let's just make sure our goggles are on and then we don't have to worry about it. Okay. But other than that, there shouldn't be any problems. Everything will be labeled and all that kind of stuff. There's nothing that should start a fire or anything like that. If you somehow find a way, wow, there really shouldn't be any way to get a fire started in there, get it all to away. All right? Okay, questions on how the lab is going to work? Okay, I don't generally make up lab groups. Okay, I mean, you guys are 15, 16 years old. Okay, I'm going to hope that everyone can include everybody and make effective groups that can get the lab done in a reasonable amount of time. Okay. When we're all done, everything gets rinsed in the sink and returned to the cart at the front, okay, and then everything's good to go. Okay. When we're done, we should have time to come back here and talk about the analysis and the conclusion and all of that, but tomorrow will be all about setting up all of the pre-lab stuff and doing that, okay? All right, I would say that right now, let's figure out our groups and maybe start getting a bit of a procedure together on paper with each other. Okay, that's what we should be working on for the remainder of class. Getting kind of a rough draft of a procedure of these steps that we've gone over here. Okay, a rough draft, and I'll go into it in more detail tomorrow.
How many people per letter?
Let's say maximum of 4. Okay, if we have more than that, there's not it's not as effective around the stations. Okay.
Exactly.